them, turn them into me so that I can get the thing. A lot of people, everybody's doing really good about putting the class. I haven't had to look in both sets of grades to find out what class you're in. So we're going to talk about motor units, okay? A motor unit is the motor neuron, so that's the nerve that's coming out of the central nervous system, and all of the muscle cells it innervates. It could be a few muscle cells for fine coordination, okay? Or it could be a lot of muscle cells for a big muscle. But a motor unit is defined as the motor neuron and all the muscle cells it stimulates. A branch to spread an impulse to more than one skeletal muscle cell. So there is no motor unit that only goes to one muscle cell. In areas of fine motor control, which are the fingers and the eyes, okay, especially in the eyes, because you have 6 muscles in the eyes, and you need very, very good coordination because it doesn't take anything. It didn't take something off very much for you to get double vision. Okay. You got one motor unit innervating probably 10 muscle cells, which is a very small thing. Okay, so it's for very fine. It's also in the hands. Okay, more powerful muscles, you got one neuron per 500 muscle cells, and so the strength of any one particular muscle contraction is controlled by the number of muscle cells per motor unit. So if you stimulate a nerve and it's to something that's regulated with fine muscle coordination, you're not going to generate a whole lot of tension. Okay, you're not going to generate a whole lot of tension. Whereas if you had a motor unit that innervates 500 muscle cells, and remember, a muscle cell is made up of myofibrils. Okay, so a muscle cell is not an individual myofibril. It's a bunch of myofibrils, a bunch of filaments. Okay, but you have a motor unit that innervates 500 of those. When that gets stimulated, then you're going to generate more tension in that muscle as opposed to one of the fine motor unit muscles, okay? And this is sort of something that, that's fashioned along that. If you got a small fiber motor unit, okay, got a small fiber motor unit and that fires, in other words, the signal comes down. And the thing about the muscle is either all or none, okay? If it gets the signal, and the signal is threshold. In other words, the signal is enough to cause the depolarization to occur in the muscle cell, then it's going to contract, okay? And so in a fine motor unit or something with small fibers, we have developed this much tension, okay? If we stimulate motor unit number 2 in addition to the motor unit number 1, okay, now we have developed this much tension. And then finally, if we add motor unit number 3, okay, we have developed this much tension. And if you think about it, sometimes we need to extend the knee. We use the quadriceps complex to extend the knee, but we're not extending it against resistance, okay? So we're firing probably small and medium. I'm just, I don't know. I'm just making this up, but we're probably firing small and medium because we're extending it against gravity, okay? As opposed to if you're at one of the gyms and doing leg extension exercises, you're extending your knee, okay? And as the weight goes up, you are recruiting more and more fibers to develop tension to move that weight until the weight is so great that you have developed tension, but you're not moving the weight. And we'll talk about that in a little while. But we have all these different kinds of fibers in every muscle because we don't, if we're picking up a glass of water as opposed to if we're picking up a kettlebell, we need different numbers of fibers to be recruited in our biceps muscle. Stimulation one motor unit will cause a weak contraction of the entire muscle. Many motor units cause a stronger contraction. Motor units conform to the all or none law. We'll also see that in the neuron, okay? Once the neuron reaches threshold. Now, this is into chapter 10. Chapter 11's got a lot of physiology in it. Notice that once we get to the nervous system and special senses and the endocrine system, we go back to one chapter at a time. Okay, we go back to one chapter at a time. Skeletal system and muscular system, we go through, you know, 2 lectures each, because one of the chapters is actually what the lab was going to be. So we don't have to sit there and me point a pointer at a muscle and say this is pectoralis major muscle, because you can look at the picture just as I did. But a motor unit is either all or nothing. It's either going to fire or it's not, and it's going to fire. It's going to generate the maximum amount of tension that that motor unit can generate. Okay, so it is going to contract or it's not going to contract. Muscular system terms: we have a twitch, a response of a whole muscle to a single stimulus. Okay, so you get one stimulus. And so if we were doing a physiology lab, and so if you take physiology as a separate, you probably will do some type of frog leg preparation where you isolate the nerve. You'll put a stimulating device on the nerve to the frog leg and stimulate the leg, and you will get a twitch, okay? And you'll have a piece of string attached to a tension meter, and you'll do a recording during that period of time. But if you get a single response to a whole muscle, it's a single stimulus, and you have a latent period, in other words, a period that it takes, and that's when all those biochemical things are happening, a contraction phase, okay? That is when the muscle, remember we had the actin and myosin, don't forget the actin's gonna slide over the top of the myosin. Actin's gonna slide over the top of the myosin. And don't forget about the troponin and the tropomyosin. Okay, these are all things that you'll be quizzed on. Okay, and obviously it's different in your case because you'll have an open book thing, but you could very well have a short answer question about troponin and tropomyosin and the acting molecule. Okay, so we have a latent period, we have a contraction phase and a relaxation phase. For skeletal muscle contraction, it's 7 to 100 milliseconds for the entire process. Okay, how much is a millisecond? Okay, how much is a millisecond? That's 1,000th of a second, so that's 10 to the minus 3 seconds. Okay, and if it takes 7 to 100, then that would be 100 times 10 to the minus 3. And so you could actually, convert that if you wanted to convert the thing. But if you just wanted to report in milliseconds, okay, that's milliseconds. Microseconds would be 10 to the minus 6. Picoseconds would be 10 to the minus 9. Okay, excuse me, nanoseconds would be 10 to the minus 9, and picoseconds would be 10 to the minus 12. You just need to get in your mind these 10 to the minus 3, 10 to the minus 6, 10 to the minus 9, and 10 to the minus 12, because you're going to be using those through the rest of your life. Because I told you nanotechnology, genetic manipulation, and things like that are going to be very, very big, okay, and things that you're going to do if you go into the healthcare field. Okay, so short period of time for skeletal muscle contraction, very short period of time for skeletal muscle contraction for the entire process, okay? So this is what a muscle twitch looks like, okay? This is what a muscle twitch looks like. We have a time element here, and then we have a generation of tension, okay? We have a generation of tension. We could also have a muscle length, where this is 100% muscle length, and then we could look at the length as we generate tension. As we generate tension, if we can overcome the weight, then the length should shorten. But on this one particular graph, we have time, okay, on the x-axis, and on the y-axis, we have percent of maximum tension, okay? And so we have this latent period, we have a period of contraction, and this is the peak of contraction, and then we have a period of relaxation. We have a refractory period, brief period early in the twitch, when the muscle cannot respond to a second stimulus, okay? What that means is there is something going on internally, that are the mechanisms are being reset. And we don't define it. We don't define it in muscles like we do in nerves. And when we get to the chapter 11, we'll talk more about that. But there's a period of time in which it doesn't matter if you give another stimulation, the muscle is not going to contract, okay? So it's not going to respond to a second stimulus. For a skeletal muscle, it's about 5 milliseconds. For a skeletal muscle, it's about 5 milliseconds. It's longer in cardiac muscle, okay? because if somebody had a ectopic focus, okay, normally the heart beats 72, 70, 80 times a minute. It can beat faster, based on sympathetic, parasympathetic innervation that we'll talk about in chapter 18 when we get to Anatomy and Physiology 2. And we'll talk about some when we talk about the autonomic nervous system, but they are limits to how fast the heart will beat based on the regulation. If we had an ectopic focus and there wasn't a refractory period when the muscle would not contract due to a second stimulation, we could get the heart beating so fast that it wouldn't have time to fill. Okay, that wouldn't have time to fill. That's the basic purpose of the refractory period, is allow time for the atria to fill and then for the ventricles to fill. Okay, that's why we have that refractory period, and we have some other mechanisms that you'll learn in chapter 18. And so it's much longer in cardiac muscle, because if the heart doesn't fill, then we're not generating cardiac output. If we're not generating cardiac output, we're in a particularly bad situation for the organism. Okay, we can if we are in the relaxation phase. Okay, if we are in the relaxation phase, remember, muscle is going to contract or not contract. Okay, so back here, once we go out of the refractory period, we can put another stimulus going there, but all the muscle can do is contract. Okay, all the muscle can do is contract, and it's going to contract as fast as it can contract. It has a set speed. Each one of the muscles has a set speed as to what the refractory period is, what the contraction period is, and what the period of relaxation is. Okay, if we set a second's twitch here, what would it do? Okay, it's already going to contract up to the maximum force it can generate. Okay, it's already going to contract up to the maximum force it can generate, so it doesn't add anything to, we can have a second twitch there, we can have a third twitch, we can have a fourth twitch, but it's not going to make a superhuman contraction. Okay, it's not going to make a super contraction. But if we go into the relaxation phase, okay, if we go into the relaxation phase, at that point, we can give a second twitch and a third twitch if we want to, and we can sum those up. It's called temporal summation. Temporal means over time, okay? We can sum those up over a period of time, and we can actually get the muscle. We can either fatigue the muscle to where it won't contract at all, or we can get it into tetany where it's in constant contraction. But here's what, so we can add a twitch, a twitch, and then start summing up these muscle contractions. And we eventually can get into tetany where there's no relaxation, although that's very rare. Usually what happens is the muscle quivers or fatigues. I mean, if you've been in the gym or been a runner or anything like that, which I used to go to the gym. I've never been a runner. Well, I was a runner a long time ago, but that was in grammar school when we played football. But you can get the muscle to quiver if if you were doing that leg extension machine and you did it to exhaustion. Okay, but this is an example of tetany. Okay, we've applied multiple stimuli and we've summed these stimuli up till we've gotten up to this contraction. Okay. Because skeletal muscle contracts so rapidly, it can undergo tectonic contraction routinely. It may have to repeat it, be able to respond any further. Okay, and so it is. In other words, you can wear your skeletal muscle out. In other words, you cannot, if you're doing bicep curls, you cannot do one more bicep curl. Okay, you've started the heavy weights, you've worked your way down, and now you got the 1 pound. You got the 1 pound weight, 1 pound. Dumbbell, and you cranked out a few contractions, and you cannot even get the muscle to do one more flexion. You can't get it to flex one more time. Okay, and it is the only type of the three types of muscles. Remember, we have three types of muscles. We have skeletal, we have cardiac, and we have smooth. It is the only one that fatigues. Okay. Remember, we don't want the cardiac muscle to fatigue, because the cardiac muscle, we got to have it to work, 70, 80 times a minute, every minute, for as long as we're alive. Okay. And so skeletal muscle is the only type of muscle that fatigues, And it's because of the system here. Muscle tone, what's muscle tone? Well, under my 40 pounds of fat, I'm sure I don't even have this either, but for some of you, this is when you have a partial contraction, partial sustained contraction that keeps the muscles in tone. We actually all have this, okay? At any one point, some fibers in that muscle are contracting to keep the muscle available, keeps the muscle in tone and available for contracting, okay? And so if somebody has a low percentage of body fat, low percentage of body fat, very low percentage of body fat, then you can see that tone in the abdominal muscles, okay? But you can see tone. You can see tone yourself because you have muscles that are constantly not partially contracted, but minimal contractions just to keep the muscle toned up, because it's easier to start, I guess, from a position where it's not completely relaxed, but is partially toned. That's why if you see most athletes, they're all the time bouncing on their feet, okay? Baseball players are bouncing on their feet if they're in the infield or outfield waiting for the pitch to be delivered. Same way with tennis players. Golf players are not doing that, but they got their little twitches too. And so this is exhibited by all healthy skeletal muscle, except in cases of stroke or Bell's palsy, okay? We have two types of muscle contraction. We have an isometric and an isotonic. At some point in my lifetime, I forget when it was, isometric was the big rage, okay? It is an external load that prevents shortening, but you generate tension, but the load doesn't move. So I've got, if you could see it, I've got a huge roll top desk here that I had made for me when I was in Louisville, so some 40 something years ago, okay? I could put my hands underneath there and try to lift that desk up, and I can't lift the desk up. I have to take it apart to move it, and then I can move the individual parts, okay? You've done the same thing. You can push against a wall. You can push against the wall. You can develop maximum tension in that muscle. You can develop maximum tension in that muscle, but the muscle will not undergo any shortening, okay? If you are somebody who does weights, okay, if you're somebody who does weights and does them fairly extensively, you will get somebody to spot you, okay? Say if you're doing bench presses or even curls or anything else like that, to help you through the last 2 or 3 attempts to do whatever you're trying to do, okay? Reps. I couldn't think of the word. That's what happens when you get to be 70 years old. I can't think of the word. The last reps. And so part of that, say you're doing a bench press and you're pushing against the bar, but you are not moving the bar. So that is an isometric contraction. Before you were moving the bar, that was an isotonic contraction. But now you're not moving the bar, so that's isometric. You are still developing maximum tension in that muscle, but the muscle, because of that particular thing, is fatigued. In the case of my desk, it is a load that those particular muscle fibers cannot overcome. Now, if I was able to get underneath my desk, okay, and had some kind of thing that I could balance on my shoulders, I probably could lift the desk up because I could use my quadriceps muscles, okay? But using my biceps muscles, I can't lift the desk up because that won't move this desk, okay? And so this is an example, okay? This would be a muscle test, and there would be a nerve coming out here, and you could stimulate the nerve, or you could just put an electrode right here and just stimulate the muscle, okay? But if you stimulate the nerve, it would pick up all the fibers because there is a nerve, okay? There would be a nerve coming to this muscle, which would have multiple motor units in that nerve. And so you stimulate this muscle has generated maximum tension. Okay, so here's a period of time we've undergone a stimulation. Okay, so this is the amount of tension, okay, that the muscle has developed. This is the amount of resistance that this weight provides. The tension that this muscle can generate, the tension that this muscle can generate will not overcome this weight here. Okay, so if we look at only this graph, okay, we look at this, we had stimulation, okay, we had a latent period, we had a period of contraction, continued stimulation, because this is skeletal muscle, we can continue to stimulate it, and we had a period of relaxation. But if we look at the second graph, which is this muscle length, so here's 100% of the muscle length, here's the same time frame. We generate maximum tension. We did not change the length of the muscle. It's still at 100% length. And so this is an isometric contraction. So this was the way to get strong, okay? You got a book, you got a video, you probably got a VHS tape back then, okay? You probably got a VHS tape. If they had, yeah, they probably got a VHS tape. Or Betamax. Remember, Sony came out with Betamax. Betamax just never took off. But this is isometric, as opposed to an isotonic. That's the one we're most familiar with. Okay, we've got that dumbbell in our arm, and we're doing a biceps curl. Okay, we've got that dumbbell in our hand. So we get actual muscle shortening occurs. The tension that your is able to generate can move the load. Okay, so here we go. Same preparation we had before. Okay, we apply the stimulus. Notice that this muscle shortens, okay, and the load is moved up. So if we look at this graph here, and this should be the same graph, but it's not. If we look at this graph here, it might be the same graph. But what it says is we have had a stimulus, we had a refractory period, we generated tension, okay, and then relaxed. Well, look what happened here. We had a refractory period. We started generating tension. As we generated tension, we had a decrease in the length. Notice we didn't go to zero, okay? We didn't go to zero because there's only a certain amount that was acting, and myosin filaments can slide over the top of each other, okay? We didn't go to zero, but we did shorten the muscle. So this is isotonic. So make sure you know the difference between isometric. Okay, isometric is you pushing against the load, generating maximum tension in your muscle, but the load does not move, so the muscle does not shorten. Isotonic, you pushing against the load, the maximum tension is generated, you are able to move the load, and the muscle shortens. So that's a good differentiation. Okay, so I alluded to the fact that there's a period of time that skeletal muscle can contract, and it's very short. And I also told you that cardiac muscle cannot fatigue. Skeletal muscle is the only of the three types of muscle that can fatigue. Cardiac muscle doesn't fatigue. Smooth muscle doesn't fatigue. Cardiac muscle, contraction, prolonged action potential and contraction. Total time for contraction is 0.25 to 0.3 seconds. 2 to 2.5 times longer than skeletal muscle. Remember, skeletal muscle was 6 to 100 milliseconds. Now we're talking about fractions of a second. The refractory period in cardiac muscle, so it's not going to contract, is almost equal to the contraction time. So the cardiac muscle will not respond to a second stimulus until after relaxation is complete. Okay, so what happens is the atria fill up, they contract. While they're contracting, the valves open up. Excuse me, the valves are already open, and blood flows into the ventricle. Now the atria are going to relax, the ventricles are going to contract, the valves are going to close, and the right ventricle is going to send blood to the lungs, and the left ventricle is going to send blood through the aorta throughout the body. And you're going to learn this in great detail when you get into anatomy and physiology too. I'm just giving you an example why we need a longer refractory period, okay? Because we need to fill the heart up. We need to fill the heart up. The atria need to fill up. And then the ventricles need to fill up. Certainly the ventricles need to fill up because we need to move that blood out to the lungs from the right side and move the blood out through the body on the left side. So remember that the cardiac muscle is you don't need to remember the times. What you do need to remember is 2 to 2.5 times longer, okay? Skeletal muscle is very short, very short. Skeletal muscle can fatigue. Cardiac muscle is its contraction and relaxation times total time is 2 to 2.5 times longer than skeletal muscle, and that its refractory period or its rest period, refractory period, is equal to its contraction time. Smooth muscle is even slower, 2 to 3 second contraction, relaxation time, no tetany, no fatigue. Think about what smooth muscle. Smooth muscle does a couple of things, okay? The big things I think about. Number one, there's smooth muscle around the arterioles, the capillary beds, small arterioles, capillary beds, and venules, okay? And they're regulating flow, okay? They're regulating flow. There's smooth muscle, 3 layers of smooth muscle in the stomach, and 2 layers of smooth muscle in the remainder of the GI system to mix the food and to move it along through the GI system, okay? Well, you don't have to be in no great hurry for that, for either one of those things. They don't happen instantaneous, okay? So 2 to 3 seconds contraction, relaxation time, so no tetany, no fatigue, okay? Energy and muscle contraction. How does muscles contract? Well, we need ATP. If the answer is if it's active, if it's anything that's energy related, we're going to need ATP, okay? And we're going to need ATP. Right now, you and I are using billions of units of ATP, okay, for meditation, for thinking, and then we're having other millions of units if our GI system's working, whatever else is going on, our heart's beating, so we need a lot of ATP. Well, guess what? We don't have but a few seconds worth, okay? So it provides energy for cross bridges, detachment, calcium pump. We're talking about in regards to the muscle, okay? But I just told you need a lot in the brain, you need a lot in your muscles, you need a lot of other places. So we got to have a lot of ATP. In the muscles, we only got a few seconds worth, okay? Well, what's one way that we can generate ATP, okay? Well, if we have this compound called creatine phosphate, if we saw this compound called creatine phosphate, we can convert creatine phosphate into creatine, and in the process, we can take adenosine diphosphate, transfer that phosphate over to the molecule and make it ATP, okay? Make it ATP. So there again, if you're a person who reads the muscle magazines, where it be Men's Health, Men's Fitness, Muscle World, whatever they are, okay, whatever they are, the muscle magazines, all you see, the big ads about creatine. You can buy creatine in all the health food stores. You can buy creatine in all the gyms. Okay, you can order out of the muscle magazines. You can order out of the fitness magazines. And the philosophy behind it is this is what it does. It really doesn't do that very much, okay? But all you have to do is explain to somebody. All you have to do is explain to somebody that this is what's going on, and you can sell them a $20 bag, a $20 thing of that stuff, okay? It does work to some. I mean, it will increase your strength. There's no doubt about it. But you can't recharge all of your ATP, okay? You can't recharge all of your ATP. You're still going to need cellular respiration. You're still going to need cellular respiration. Oh, as an aside, the absolute best recovery drink post-exercise is not something you buy in the gym. You go to Gate, you go to Publix, you go to Winn-Dixie, you go to Walmart, you buy you 2% chocolate milk or 2% milk, okay? You get the perfect combination of carbohydrates, proteins, and lipids for a recovery drink for 99 cents, okay? For 99 cents, as opposed to a $5 protein shake that you buy from the gym or wherever you go to do the things, okay? So I'm telling you, if you want the absolute perfect recovery drink for right after you exercise, this is not for your meal later on or whatever. Okay, this is for a recovery drink for exercise. Yeah, the thing is, I drank 2% chocolate milk at breakfast, but it's 2% chocolate milk, and the people who are serious about doing this will tell you that they don't buy that other stuff. And for once that all that information came out in the magazines, you couldn't get 2% chocolate milk in any of the grocery stores because everybody was buying it out. But just remember that. That's on the side. Yeah, that's free information, okay? All right, so we have a few seconds of ATP, okay? We may be able, with loading this stuff, to give us another 14 or 16 seconds, but guess what? If we don't have cellular respiration, we're just not going to make enough ATP. Cellular respiration is the opposite of photosynthesis. Photosynthesis, you take CO2 from the atmosphere, a green, we'll just say a bush or a tree, takes CO2 from the atmosphere, takes water out of the ground in the presence of sunlight. Sunlight provides photons and converts that to glucose and oxygen as a byproduct, okay? That is photosynthesis. Well, what do? We take that glucose in the form of starch, okay, or in the form of glycogen, one or the other. We have either stored it or we're going to eat some. We're eating plants or we're eating the animals that ate the plants. Either way, we're going to break glucose down in the presence of oxygen. So we've got to have glucose and oxygen. That's going to give us CO2, water, and energy. And that energy is going to be in the form of ATP, okay? So this is a little schematic. We have in Anatomy and Physiology 2, there's a whole chapter on this, okay? Whole chapter on glycolysis, carbohydrate metabolism, lipid metabolism, protein metabolism, and nucleic acid metabolism. There's one whole chapter on metabolism after the digestive chapter. So you'll get this in much greater detail, okay? But we take in glucose, and in the cytoplasm, glucose is a 6-carbon molecule, as you may or may not remember from chapter 2 chemistry. We break that down into pyruvic acid, 2 3-carbon molecules, okay? We break that down into pyruvic acid, 2 3-carbon molecules. If we have oxygen present, then that goes into the mitochondria. So this first step occurs in the cytoplasm. If we have breaks glucose down into 2 3-carbon molecules. Glucose is 1 6-carbon molecule. Breaks it down into 2 3-carbon molecules. That's pyruvic acid. This then goes inside the mitochondria, okay? And we're going to form CO2. We're going to form H2O and 32 net gain units of ATP. So you can see this is a much more efficient system, for making ATP. If we don't have enough glucose, if we're in a glucose deficit, then we can take fatty acids. Fatty acids, remember we have a triglyceride, which was a glycerol backbone and 3 long-chain fatty acids. Well, we can break that down and we can take the fatty acids and move them in here inside the mitochondria, or we can take amino acids. In medical school, I took an elective, a 2-credit-hour elective, either in my first or second year, probably in my first year, called amino acid metabolism, and that's all we looked at was how the amino acids got broken down. But this is what happens. If we have oxygen present, we can do cellular respiration. Okay, if we have oxygen present, we can do cellular respiration. And rather than get 1 unit of ATP out, okay, now we've gotten 32 units of ATP out. If we have exceeded our demand for oxygen, okay, in other words, our muscles are going to start to cramp and they're going to start to quiver, okay? We then go to aerobic respiration, okay? So that pyruvic acid, which has the big potential for forming a total net of 32 ATP, is not going to be able to go into the mitochondria because we are in the oxygen deficit, okay? It is then going to go to lactic acid, and we're only going to get a net 2 ATP out, okay? So instead of getting 32, we only get 2 out. Now, fortunately for us, when we quit exercise and take a few deep breaths and overcome our oxygen deficit, our temporary oxygen deficit, then this lactic acid, which is in the circulation now, will be converted back to pyruvic acid and we'll be able to use it. Okay, so it's not like it's lost forever. But so this is the importance. So we have a little bit of ATP hanging around for a few seconds. We can, if we take all the creatine phosphate that we have in our body and dump one of the phosphates so that it becomes creatine, we can make ATP. So that gives us another 14 seconds. If we have oxygen present, we can get all the ATP. And if we have oxygen present and we have nutrition, okay? If we have oxygen present and we have nutrition, then we can make as much ATP as we need. I said this to the other class because we just did the metabolism chapter. Okay, we just did the metabolism chapter, and we're going to go to the urinary system. Under no circumstances do you want to be on anything like an Atkins diet when you're taking an exam. The brain needs glucose. The brain needs glucose. If you don't fulfill the brain with glucose, it will burn amino acids and burn lipids, but the problem is they're not as efficient, okay? And it takes, some other steps have to go during the period of time. So under no circumstances do you want to be taking an exam and be in a glucose deficit. In fact, instead of drinking your Diet Coke or your Diet Coffee with your sweet and low or whatever you put in your coffee, okay, you probably would want to do a little bit of glucose loading before you had your exam. That's just a tip. And it probably, it's a tip for you for these regular exams. It's a tip for you. It's certainly a tip for you. It's certainly a tip for you when you take your board exams, okay? This is certainly a tip for you when you get ready to take your board exams and any kind of certification exams. Under no circumstances do you want to be under a glucose deficit, because you've got to have glucose for your brain to function maximally, okay? So this is what happens if we don't have any oxygen. We're only going to get 2 ATP. If we got oxygen, we're going to get 32 ATP. We're going to go to pyruvic acid, and then we're going to go to lactic acid. But fortunately, when we overcome our oxygen deficit, that's going to be converted back, okay? Causes a temporary burn sensation. Everybody's experienced that, even me. At some point, oxygen debt must be restored and waste products removed or converted. So that's what's going to happen there. Velocity and duration of contraction. We have different types of muscle fibers, okay? Guess what? There are some people that can do endurance things, okay? There are some people that can do endurance things. I mentioned grammar school football. I was the kid that could learn, that could do the laps, okay? I was the kid that could do the laps. I could just run and run and run and run, or I could swim forever. Not forever, but I've got a lot of endurance fibers. But wind sprints kill me, okay? Because I don't have a lot of the fibers for they're called fast twitch fibers, okay? And those are your sprinters, okay? Are those your tennis players? Are those your golfers that hit the ball 300, 350 yards? You got to have a lot of fast twitch fibers to do things like that. So the velocity and duration is based on the muscle type of fiber, the load, and the recruitment. We have slow oxidative fibers. They are aerobic, they resist fatigue, have a high endurance, and they're first to be recruited. So that's me. I can do something, just keep doing it. And an example was, I was the kid that, if the coach said us to run 10 laps after practice, I said, okay, that's fine. Or 10 laps before practice, didn't matter, whatever it was. Well, whatever it was, I could do that. One of my few talents, okay? So I had a lot of these slow oxidative fibers, and a lot of people just, they have predominant of one type of fiber. So these slow oxidative fibers, they are aerobic, as opposed to the fast glycolytic fibers, okay? They are anaerobic. If somebody's got their microphone on, they need to cut it off, please, unless you're going to ask the question. Fast glycolytic fibers are anaerobic, okay? They are fast to fatigue. They have very low endurance. They're the last to be recruited, good for short-term, intensive muscle requirements. Short-term, intensive muscle requirements. Those are your 100-yard dash folks, okay? Your 100-yard dash, your 50-yard dash. Those are the people that can knock the fire out of the golf ball. You know, my swing speed or my ball speed is like 80 miles an hour. Their ball speed is like 140, 150 or whatever. It might be faster, I don't know, okay? I hit it maybe 150, 180 on a good day. They hit it, you know, Bryson DeChambeau hits it 300, 350. He caught the cart path the other day, hit over 400 yards. But they have a lot of fast twitch fibers. They can swing, and it's, you know, they're only doing it one time, okay? And so they recover from it. Are the 100-yard dash guy is going to run a race, okay? Or guy or girl is going to run a race, and then they're going to be done for that day. Now, training is a different thing, okay? But usually they're going to run, maybe they might run in morning heat and evening heat, but they're only going to run 200-yard dashes, okay? They're only going to run 200-yard dashes. And so those are anaerobic fibers. You've got enough ATP to do that, okay? You've got enough ATP, and those are the fast twitch fibers. And those are good for short-term things, and you might be one of those people, okay? I'm one of the other type of people. We have all the fibers in our body. We just have, different people have predominant different fibers. And then we have these fast oxidative fibers. So we have the slow oxidative fibers, the fast glycolytic fibers, and then the fast oxidative fibers. That means oxygen, okay? Slow oxygen. Fast glycolysis, so we get to pyruvic acid, and then we go to lactic acid, okay? And then the fast oxidate, we're using oxygenating, or we're using it in a little bit more efficient manner. So these folks are somewhere in between, okay? Effects of exercise. We do not create new muscle cells, but we do create new muscle fibers. And the muscle cells can hypertrophy, okay? Everybody in this class knows that if you want to make your muscle big within certain restraints, you can do that by exercise, okay? Increased efficiency of respiratory system gas exchange becomes more efficient. So these are the benefits of exercise. We don't create new muscle cells. We can increase cardiac output. The heart hypertrophies. It pumps more blood out on each beat, and it pumps it at a slower rate per work demand, okay? And so if you're somebody who's a endurance athlete, or maybe not even an endurance athlete, you're going to get hooked up to the machine that's going to measure your gas in, gas out, measure all these things, and going to look at you, and you're going to have EKGs and blood pressure monitors and everything like that, and they're going to see what your efficiency is, to try to tailor your training schedule, so that it can maximize your output for what your individual sport is. Maximize your output for what your individual sport is. We also train, I think I mentioned this before, but the Olympic team, part of the Olympic team trains at altitude, because when we went to the Olympics in Mexico City, it was 5,000 or 6,000 feet, and we were training, at sea level, or 500,000 feet, and we were at a significant disadvantage. So they moved the training facilities to I believe in Colorado Springs, if they still train there, so that you could train at altitude, so you could develop increased efficiency at decreased oxygen levels, so that when you got into an environment that was sea level, or 1,000 feet above sea level, and you had increased oxygen, that could increase your efficiency, okay? In the old days, you used to do blood doping. They would give you would take units of blood so that you carry more oxygen. to increase your to increase your performance. Skeletal muscle hypertrophies, increase in muscle size through the adding of fibers and increased mitochondrial numbers. So we don't add individual muscle cells, but we can hypertrophy the fibers, okay? And obviously somebody who is bathed in testosterone, okay, bathed in testosterone can hypertrophy them better. So if you're females, as a general rule, don't build huge muscles. They can get great tone and they can get great strength, but they don't build huge muscles because they lack testosterone. Males, especially young males, okay, can build larger muscles because of the presence. This is just an endocrinology thing, because of the presence of testosterone. So for the younger people in the class, you can build bigger muscles than I can at 70 years old because my testosterone level decreases with time, okay? Now, if you want to build big muscles, whether you are a male or female, all you have to do is take exogenous testosterone, which is what all the bodybuilders do, okay? But it has a number of bad sequelae with it, and so that's not anything I'm advising, but you can, whether you're male or female, increase your muscle size by taking exogenous testosterone, but it's not anything that I would advise anybody to do unless you have a testosterone deficiency. Then you'd want to do that. There are a number of disorders affecting the muscular system, and had we been a face-to-face, we might have used one of the labs to do a roundabout teaching thing where I gave you had four partners. We had a lab desk usually had four people, or had four people, and each one of you have a different part of the case scenario and present it to everybody and then come up with a possible diagnosis. But you can have a disturbance in the upper motor neuron, okay? The upper motor neuron, essentially the brain and some of the tracks going down to the spinal cord, which we will look at. We don't have to learn a whole lot of tracks, and there again, because you have an open book test, it won't be as hard, okay? There is a bunch of anatomy, so just remember that. Cerebral palsy is one of these things, okay? Usually caused at birth, okay? Usually fetal damage, voluntary muscle control is lost, doesn't get worse, but is irreversible, and occurs in about 1 in 6,000 births, okay? A cerebral vascular accident or stroke, obviously is a central nervous system problem, high up, in the brain, okay, where you have a stroke. So that would be a disturbance in the upper motor neuron. These are just two examples, okay? And then you can have multiple sclerosis, which is demyelination of the nerves. We actually have demyelination. We'll learn that the nerves have a fatty wrapping around them called myelin, okay? And that myelin allows the nerve signal to travel super fast, okay? As opposed to a nerve that doesn't have myelin, okay? So if we have demyelination of the nerves, then that is a serious thing, because if that's a nerve that needs to send the signal fast and is no longer sending that signal fast, then whatever it's supposed to be doing could result in a significant problem. So that's multiple sclerosis. So these are three upper motor neuron disturbances. And then lower motor neurons, one of the things is poliomyelitis, okay, poliomyelitis. It destroys the anterior horn motor neurons. That doesn't mean a thing to you, but there's an anterior, posterior, and a lateral horn in the spinal cord, and we'll look at that when we get into the nervous system and the spinal cord, okay? But it destroys the anterior horn motor neurons and the muscles that are served by these nerves. So if you disturb, this is where congregations of the central nervous system cells are going to congregate in the anterior horns for the motor, okay, for motor, and send nerves out. Nerves are going to go out, and they're going to go to muscles, okay? If you destroy the input to the muscle, if you destroy the input to muscle, then the muscle's not going to contract and it will eventually atrophy, okay? If you've ever had a broken limb and it's put in a cast, and it's put in a cast for 4 weeks, you can, when they take the cast off, you can see that the muscle's atrophied. Well, imagine that over a period of time, okay? Imagine that over a period of time, and then the muscle completely atrophies. If it's a important muscle like the respiratory problems, then you can develop an inability to breathe. And if you've ever seen any old movies where it's usually kids, okay, they're in iron lungs, okay, and all in iron, you remember, you don't remember because we haven't done the respiratory system, but all we do to breathe is when we breathe, we drop our diaphragm, which is one of the muscles, okay, we drop our diaphragm, we increase the volume inside of our chest. When we do that, the pressure inside of our chest goes down. And so the pressure inside of our chest is less than the atmospheric pressure outside, so air flows in. When we exhale, we relax the diaphragm, we decrease the pressure in our chest. Well, the pressure in our chest was 760, and then we've decreased, and we still have that same volume, okay, because we've decreased the volume, and we was at 760, so that means it's going to go up, so then the air goes out because the outside pressure is 760. So what these iron lungs did, all they did was create a pressure difference. They created a pressure difference because ventilators weren't that common, okay? They weren't that common. I remember I told you that, I can't remember, I think I was 8, 9, 10, 11, somewhere around in there, I do believe. We were all taken to the high school and given the Sabin vaccine, okay, the polio vaccine, because polio was a disease that you could get when I was growing up as a child. We had polio hospitals where you were sent if you had polio, and there would be just wards of these iron lungs. Now you could be put on the ventilator, okay? You could put a ventilator or have an assisted ventilator maybe, but in that day they didn't have those, okay? And then that was a disease. Well, the long and short of it is you may, as I keep reiterating, okay, for 2 reasons, okay? Number one, you're going to see people that are coming to this country that did not, through no fault of their own, have access to medical care and may not have gotten immunizations, okay? May not have gotten immunizations. So they didn't get an immunization against the polio vaccine, okay? They didn't get immunizations, get polio vaccine. Now, in the United States, at one point, we had herd immunity. If you hear the thing herd immunity, everybody got vaccinated against polio because everybody probably knew at least one person that had polio or knew of somebody in their family or somebody they knew had polio. Okay, so everybody got vaccinated. I mean, you literally went to the high school on Sunday and got the sugar cube with the polio vaccine. I think you had to go get 2 doses or something like that. Well, you're going to have people that didn't get vaccinated. So the number of cases of polio are rising every year. Okay. The second thing is you've got the parents who, for whatever reason, have decided not to get immunizations for their kids. And that's their decision, except their kids now, if polio is out there, and it could very well be out there because we have people who have polio now, then there's a possibility that their children, okay, and it doesn't matter what socioeconomic class, they could be very rich if their parents didn't get them vaccinations, okay? Parents didn't give them vaccinations, they could actually come down with polio. So all these diseases that I only read about, okay, you may see, okay, so just you always have to keep this in the back of your mind. And then obviously if it affects the medulla oblongata, which is one of the control centers. And my next big tip for you is when you're taking your certification exam and they ask you what the control center is for some automatic thing, if one of the answers is medulla or pons or medulla and pons, just pick that, okay? Just remember that the medulla and the pons are big time control centers for the autonomic stuff that goes on, okay? So 10 years from now, 5 years from now, 5 years from now, 2 years from now when you're taking your exams, and 5 years from now, 10 years from now when you're taking your recertification exams, and you didn't have a clue they were going to pop this question in on you. And it's just one of those things that you just have to remember. And if you remember that it's the medulla and the pons, so if they have both of those as a choice, pick that and you'll get it right 99% of the time. So if it affects the medulla, which is where the autonomic system is for the regulatory system for the heart, okay, to speed it up and slow it down, then you can get cardiac arrest. Okay, disturbances in the muscle tissue, you can have muscular dystrophy, okay. And I don't know, probably nobody remembers Jerry Lewis, but Jerry Lewis used to have a Labor Day telethon, your Labor Day telethon about this, MD, Muscular Dystrophy Association. What happens, you get no production of this particular protein, okay? This particular protein attaches the cytoskeleton to the extracellular matrix. So this is a scaffolding framework, okay? If the muscle is going to contract, okay, if the muscle is going to have to contract, it's going to have to contract against the scaffolding, okay? Because the myosin is going to need a firm, if you're in a tug of war, okay, in order for you to pull the other team over, you've got to have a firm footing. Well, the reason you have a firm footing is you've got your feet planted against the earth, okay? Well, if the myosin is trying to pull against the actin and the cytoskeleton is not stable, the cytoskeleton, which is the skeleton, which is the skeleton part of the cell, is not stable, it has no firm footing. If it has no firm footing, it's not going to be able to do that power move where the actin slides over the myosin, okay? So you can get this in muscular dystrophy. Muscular movements, these are just some movements. I guarantee you there are going to be questions about them. There's an agonist, an antagonist, and a protagonist. Prime movers are the agonists. What is a muscle that flexes the elbow? It's the biceps, okay? What is the muscle that extends the knee? Well, it's the quadriceps complex. You know, there's individual muscles, but what's the muscle that flexes the foot, extends the foot? That is the agonist, okay? That is the agonist. Well, if we extend, okay, if we flex the elbow, we need a muscle that will extend the elbow, because now we've got it flexed, okay? We flexed it, and now we got to move the elbow back, okay? So we have the triceps. So a prime mover is an agonist, okay? An antagonist is a muscle that opposes or resists a particular movement, okay? If the agonist flexes, an antagonist extends. So if we want to extend the elbow, we contract the triceps. So when we flex the biceps, the triceps extends, but it's essentially relaxed. Remember, one of the things that muscles can do are relax and lengthen, okay? So our biceps was our agonist, biceps brachia, and our triceps was our antagonist. Now, when we extend the elbow, the triceps is the agonist because that's the movement that we're trying to do, and the biceps is the antagonist. So it's just it's one muscle that does the whatever the muscle it is that you're working with to try to do the movement you're trying to do. That's your agonist, and you have an opposite muscle that's going to relax during that time because it's going to return that joint or body structure back to its original position. And then you have a synergist, okay? Synergy is helping, okay? When you are trying to bring a cup of water, or in my case, a cup of soda, okay, up to my mouth, or a cup of tea, the biceps brachii is the agonist, but the brachioradialis, okay, the brachioradialis is a synergist because it can help the biceps bring that particular item up to whatever cup of water, cup of soda, whatever, cup of tea, up to your mouth, okay? So it can help that. You will not have to know what an agonist and an antagonist is in regards to the muscle groups, okay? What you will have to know is what an agonist is. You know, it's a muscle that's performing the movement, and antagonist is a muscle that's opposing it, and a synergist is helping the prime mover or the agonist. The agonist is called the prime mover. It's helping the agonist with the movement. So you just have to know the definitions. You're not going to get a list of muscles to pick out and say which is the agonist and which is the antagonist and which is the synergist. Okay, naming muscles. If you probably haven't started on that, and don't worry about it. If you just got here, I'm going to do the muscle practical over the weekend. The muscle practical is going to be exactly like the bone practical. You got a muscle list. I will find, today I wasn't able to locate some good videos like I did with the bones, but I'll keep looking. There's a lot of videos out there for muscle lab practicals, but no good ones like I found before. But once you got that, you got your list. That was, and as I told you before, that was actually the word list you got for the muscle practical. You came in and your answer sheet was on, on one of the 4 spots on the table with a mannequin or a limb or something like that. These are all models, and you also had a word sheet with it attached to your thing. And I gave that skeletal muscle, and I gave that for muscle side to side. I'll give you that's the same word list you would have had if you're taking the thing. So just need to go through and look up those muscles. The slides are going to come right out of the book like they did before, and you'll have you'll have till the next the close of the next exam, which is going to be the 27th of July to complete it. So you're going to have as much time as you want to complete. But they name muscles by shape, by deltoid and trapezius, by action, flexion, extension, or adduction. Okay. Remember abduction and adduction. Abduction is moving away from the midline. Abduction, because they sound so alike, abduction is moving away from the midline. Adduction is moving to the midline. Okay. By location, intercostals, quadriceps, femoris. Okay. By attachment, the 3 extra credit questions I used to give on face-to-face practicals was name the quadriceps muscles, name the hamstring muscles, and name the muscles of the shoulder joint. And that's if we had a face-to-face practical. And I'd tell everybody in advance what they were, and there would be at least 1 or 2 people that didn't, never even bothered to look them up. There always is in every class 1 or 2 people that didn't even bother to look them up. Intercostals, quadriceps by attachment. What does that attach to? Sternocleidomastoid. Okay, well, it attaches to the sternum and to the mastoid process. Okay. Number of origins. Tricep has 3 origins. Biceps has 2. Size relationship. Gluteus maximus, gluteus minimus. Okay. By direction of the fibers. Are the fibers transverse? Are they oblique? In other words, is rectus transversus, rectus oblique. They are 3 layers of muscles in the abdominal wall. Okay. And they run in different directions. And so when you do the dissection through the abdominal wall, you can actually see the fibers in the directions that they go in, and you could separate the fibers so you don't have to cut the fibers, you step in that direction, and you keep doing that, and then you can get down to the peritoneum. You've never cut a muscle fiber. You made an incision in the skin, you separate muscle fibers, and then you open up the peritoneum, and now you're in the abdominal cavity, and so they have less trauma. And that's the end of the slides there. Okay. Does anybody have any questions for me? Yeah, the test is due tomorrow by 11:55. I'm going to extend the deadline